Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 110th seminar, and I call it a NASA Day. I came back home from 2 back-to-back trips to Singapore and Minnesota, experiencing a huge temperature difference, like 50 Celsius. It was great to meet many researchers and share our research through multiple seminars and interesting discussions. Roughly 1 week later, I will embark on another trip. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Lynn Rothschild. I'm not sure I can introduce her briefly, but I will try. She does not need any introduction because we all know her. She is a senior scientist at NASA Ames Research Center and also professor at Brown University. She was kind enough to invite me to NASA last year for a seminar, and one of my bucket lists, Moon goes to Moon, and that become basically one step closer. She is well known for astrobiology and synthetic biology, and she will be the perfect person who probably could comment on my recent experience with a huge temperature change from 30 Celsius to minus 20 something, although I am not an extremophile. Lynn, thanks so much for your amazing contribution to the scientific community, and again for the invitation last year and the virtual podium is all yours now. Thank you again.
Great, thank you. It's really my pleasure to be here. And your introduction is overly kind, because I, like probably everyone else, when you're in your lab, you feel like no one in the world knows who you are. And so this has been particularly kind, and I'm particularly excited to talk about our synthetic biology work. So let me go ahead and share my screen. And as mentioned, my background is actually unusual for this group. My background's in evolutionary biology. And so I was brought to NASA for astrobiology, which was then called exobiology in those days, and have been very instrumental in that community in forming the whole field, particularly for extremophiles. But about 15 or 18 years ago, I was redirected, as it happens when you're working for the government, to start a program in synthetic biology for the agency. And so that's what I'm going to talk about today. And can you see my slide all right? Mm-hmm. Okay, fantastic. So what I'm going to do is tell you a little bit about synthetic biology and NASA's mission, since that's very different, I think, than most of your day-to-day work. So NASA, just to remind you, is the National Aeronautics and Space Administration. And when I started this program in synthetic biology, I was tasked to see how synthetic biology could be infused into our mission. So again, we're a mission-driven agency. Agencies like the National Science Foundation are pure research-oriented. We are mission-oriented, and these are our missions. The first A is aeronautics, and so we are pioneers in aeronautics. We have the 2 largest wind tunnels in the world where I work, so that's actually our big claim to fame, are wind tunnels. But also science, and this is the space part. We use space to study planet Earth. This is the Earth Science Group. But we also go forth into space to do discoveries, of course, in our solar system and now beyond our solar system. And then finally, we have a human exploration component. And I believe very firmly that synthetic biology can aid all 3 of these major missions. But what I'm going to do is focus on that last one for the few minutes that I have, and that's for the exploration part. Now, everyone here who's watching this, I'm assuming, is a human. We all need various things to live from day to day. We need radiation protection, something we don't think about because we have this protective stratospheric ozone layer. We need life support We need food, we need oxygen, we need medicine, waste recycling, clothing, and so on, communication systems, habitats, power, heat, light, and transportation. And we still need all of those things when we go off planet. Particularly things like oxygen and gravity are no longer givens. But we have challenges that, again, most of you, particularly those of you who are thinking towards the commercial sector, would not have to worry about. The biggest thing that we have is worrying about upmass. So I don't know if you remember from the launch a few weeks ago, the majority, well, something like a third to a half of the mass of a spacecraft is burned up and jettisoned in the first few minutes of the launch. So an enormous amount of the fuel is spent just getting out of Earth's gravity well. So anytime that you can reduce the mass of your payload, and I'm saying mass because of course you're changing gravity levels, is incredibly important, and it's tied into the cost. So you can no longer have the luxury of a piece of equipment that weighs kilograms. Everything has to be reduced. It has to be able to be stored. I had a satellite mission. I had to end up storing cells dried for 3 years before we even had a launch. And then everyone expected that they would be reactivated and everything would go perfectly. So it has to be reliable. It has to be flexible. You have to worry about the power. And I've been arguing that this is actually this ultimate circular economy, because it's not like you can just walk out and go to the hardware store. Now, my argument, and I don't need to convince this group, is that the technology that I believe can solve a lot of these problems is life. Once you start to think of life as a self-replicating, self-repairing micro machine that does carbon chemistry and nanotechnology, you're really off to the races. So I would love to spend the next hour telling you about all the projects in my lab, and I don't have that luxury. So let me just give you one or two of the things that we're working on right now, just so you get a flavor of the sort of things that we're doing, but it's certainly not a complete picture. So what's the big idea? Well, for example, life is extremely good at material production. Life, of course, is a material. You think about wood. Life is used to make materials. And in that second panel, you see my good colleague, Chris Maurer. Many of you, maybe none of you, have ever heard of him because he's not a biologist. He's an architect. He's the principal architect for Red House Studios in Cleveland. But he and I both have an interest in mycotecture and using fungi to build structures by binding other materials. And there he is holding a block that he's made. And so we're now moving towards a phase 3 project, trying to use this kind of technology to build structures for astronauts on the moon or Mars. We've also already used that to make some furniture and actually the body of a biodegradable drone with an iGEM project. Life is really good at material extraction. And we've had a project for about 10 years now called biomining, where we've taken this really exquisite capability of organisms to bind metals with great specificity and use that for mining applications. I mean, you think about it, the hemoglobin in your cell is bound to iron. It's not like you wake up one morning and the hemoglobin says, gee, it would be really fun to bind cobalt today. It doesn't work that way. It's iron every day, all the time. And so that kind of specificity is something that a miner would just, dream about. And then finally, life is a scaffold for material production. And one of the projects that we have in that line, and one of the reasons I particularly wanted to share the stage with Cameron today, is a project called BioWires, which was started by a wonderful undergraduate from Brown in my lab, Simon Vecchione, whose picture will probably pop up in a moment. And we're still working together at this point, now that he's a senior postdoc at NYU. And it turns out that in DNA, if you have a cytosine-cytosine mismatch, that allows the incorporation of silver atoms with great specificity. By the way, thymine, you can get the incorporation of mercury, and Simon now has a whole library of metals that you can bind. Turns out with his NMR work, it's not a single atom, it's 3 per mismatch. And so all of a sudden, thinking about DNA as just a linear thing that is tunable, you can make a very tiny wire that's completely tunable. Finally, as NASA plans the Artemis missions, because I'm required to say Artemis in every talk, to the moon and beyond, what happens when astronauts get sick in space? Right now, they can be brought back to the Earth, but that's not going to work forever. And so we've had an idea, and maybe you, if you're watching from the UK, you've seen us written up in the tabloids. This is the only time my research has been written up in the tabloids. And I don't need to point out which project is mine. And it gives me the opportunity to say, if you want to know the details, look on page 11. But seriously, that really was about us. But the project is called an astropharmacy, where we're developing a miniaturized on-demand production system for creating particularly peptide-based biologics. We have a cast of 1,000. You'll probably recognize a couple of names on there. And this is really exciting, the idea of a very tiny production system where you can produce drugs on demand, which is, again, completely the opposite of what the commercial sector on the Earth is interested in, because large drug companies want to make money by having, millions of people with a particular disease and need a particular drug. And finally, we get back to really my roots at NASA, and that is looking at life in extreme environments. Here's a picture from one of our favorite field sites from decades ago. This is Octopus Springs in Yellowstone National Park. If you turn your head a little sideways, it looks a little like an octopus. It's about a 5 or 10-minute walk from Mushroom Springs, which is where Taq polymerase was originally isolated from. So this is a very hot spring with an unusual biota. And we have asked for years, why not raid nature's hardware store and take some of these capabilities and put them in other organisms? Start to think of life not in terms of its wonderful diversity, but in terms of a genetic hardware store. And this is a project that my Stanford Brown iGEM team in 2012 called the Hell Cell Project. And I just love that. I suppose you could also call it Life 1.5. And I have now a postdoc really running forward with that project, Garrett Roberts Kingman. Or many of you may be involved in the Build a Cell program. Those seminars are on Monday early morning for us. And that's really the whole question of why not just build life de novo? Maybe we don't need life itself to do some of these things. Maybe a new creature, a new instantiation of life would be better. Maybe something that uses titanium instead of lipids for the membrane, or who knows what. But maybe you could produce something that would be even more useful for us. So that's, in a nutshell, the exploration stick. Aeronautics, I believe that we could be getting into green aviation, working for the federal government. I'm not supposed to be competing with private industry. And of course, there is interest in the outside world on biofuels. I think that we're probably about to get into novel materials and biomimetics in the aeronautics sector. And in the science, as I said, I sit in the astrobiology division. Actually, my branch at NASA Ames is planetary science. And so we've been interested in producing exploration platforms, and then particularly astrobiology, for example, the origin of life. And what synthetic biology allows you to do that the other approaches to these sorts of questions don't allow you to do is ask the questions, what if? What if you had an organism that only had 6 amino acids? Or what if you had an organism with D instead of L amino acids? Or what if? These tools of synthetic biology allow you to start to address some of these what if questions. And so thanks very much. That's what I have to say today. Here's this amazing micro machine. It can be made even more useful through synthetic biology and perhaps overcoming the limits of carbon-based life. And so again, thank you very much for this invitation, and back to both of you. And hopefully that sets you up for the main speaker today, Cameron.
So Lynn, exciting. Your talk is always fascinating. That's what I find all the time. So thank you so much. This is absolutely great. And then I also now realize, the EBRC, the Engineering Biology Research Consortium, actually currently working to, basically lay out the space roadmap. This year, we already met San Diego early, I think a couple of months ago. So we're gonna rely on you, to get some more feedback and more insight.
Yep, I will be there in March. I just couldn't make it into San Diego.
Okay, Houston, right?
Yep.
Right. Thank you so much. That's fantastic. All right. So now the main speaker of today with a longer introduction. Dr. Cameron Glasscock is a WRF postdoctoral fellow in Dave Baker's lab at the University of Washington in Seattle. His research focuses on the development of computational approaches for the de novo design of DNA binding proteins with applications in gene regulation and gene editing. Prior to his postdoctoral training, he was an NSF graduate research fellow in the labs of Julius Lux and Matt DeLisa at Cornell University, and a visiting scholar at Northwestern University. I believe that is because of Julius, moved to the Northwestern. We had a very great time at multiple conferences recently, and I see his great potential to become a future leader. And I would say, if you are a faculty search committee member, you do not want to miss him because he is one of the strongest candidates this year in the job market for the faculty position. And I heard he already had multiple interviews, also many, waiting for his, interview. So don't wait. I mean, you will lose him, to another big university if you just wait. So Cameron, thanks so much for your time today, even though you are on the road. And please take it away. It's all yours now. Thank you.
Excellent. Let me share my screen real quick. Let's see. Is everyone able to see okay?
Yes.
Great. Well, thank you very much for that really nice introduction. And I'm really excited and honored to be able to share my work with all of you today, and also to share the stage with Lynn, who has presented a really exciting big idea of kind of the big goals of what we can do with synthetic biology. And so today, I'll be kind of zooming in to a much more fundamental level of how we can start to design using computational tools, DNA binding proteins. And so I think in general, protein-DNA interactions don't need to be motivated too much. Everyone knows that they're very fundamental to many different processes in biology. But with this slide, I really just wanted to point out that although we do have tools for recognizing specific sequences, things like zinc finger proteins and CRISPR systems that are RNA guided, there are many things that we still cannot design or engineer from scratch related to protein-DNA interactions. So these are things like allosteric transcription factors, where we don't have yet the ability to have a transcription factor that would bind any DNA target site or promoter sequence in order to regulate it through a small molecule induction. And we have many different enzymes working on DNA substrates, and in some cases, we'd like to have the ability to design from scratch new enzymes, like similar things to like these homing endonucleases, but might have very different properties. And when we look at nature, we also see that it's doing many more things beyond these sort of simpler interactions and enzymes. You have things like these FOXP3 transcription factors, which form these higher order oligomeric assemblies on DNA satellite repeats that actually bends the DNA around itself. And that's important for its transcriptional regulation. And you also have proteins that are not just interacting with B-form DNA, but also many different types of DNA structures that can occur in cells. And so here's just one example of the human POT1 protein that binds telomeres at these double-stranded, single-stranded junctions. And that's important for helping to protect these sites from unwanted DNA transactions. And you also have things like these CRISPR-Cas integrase systems, which are able to integrate new spacer sequences. And part of that is mediated by the ability of some of these proteins to induce these specific U-shaped bends in the DNA. And so all of these are the types of things that we don't necessarily have the ability to design from scratch today. But as a postdoc, I've been really thinking about how can we start to develop the tools for computational design in order to do these things. And so I'll start off by kind of giving a big overview of how I'm thinking about this and how we can approach it with computational protein design, and spend most of my talk talking about the results of a project I've been doing as a postdoc in order to solve the very first simple problem of designing really small mini proteins that can bind specific DNA sequences, and then go into sort of my future directions and conclusion of where we can continue to go from here. And so starting off, I really believe that de novo protein design offers a really complementary solution to a lot of the natural tools that we have, like CRISPR systems and like zinc fingers, in order to interact with DNA and open up some of those new possibilities. And so we've seen in recent years how this set of tools has become really powerful for many different applications, things like designing protein binding or small molecule proteins that have really high affinities and selectivities for their targets. And beyond binding, we see how we can design now proteins that can change their conformations in response to things like peptide binding. We can create large assemblies of proteins that can serve as nanoparticles for display of antigens in vaccines, as well as in some cases, we're starting to have the ability to design de novo enzymes from scratch. But as I started my postdoc, of course, one thing that was kind of missing and we hadn't seen much progress on is the ability to design protein nucleic acid interactions or DNA binding proteins. And so that's what I really wanted to go after. And so as we think about developing tools in order to design protein nucleic acid interactions, it's helpful to understand how these protein design processes works. And in my view, there's really 3 steps of computational protein design, where we start with the process of backbone sampling, where we generate and sample from large sets of backbone structures. And these are really just shapes of proteins prior to having any particular amino acid sequence. And we're looking for things that have different properties. If you think about the case of designing a protein binding protein, you want to have a protein that not only has a physically reasonable fold, but also has things like shape complementarity to a target. We then go through a process of sequence design, generating an amino acid sequence that would actually fold into that protein and have different functional properties. In the case of a binder design, you'd want to design an interface with different interactions. And here, I would just point out that many of the best successes of protein design have been when we are designing these hydrophobic interfaces, which have more geometry-independent interactions. And you don't yet have to deal with what is harder for the computational modeling tools we have of polar interactions. And so that's one of the limitations that we've had in protein design until very recently. And finally, we then want to go through a process of in silico filtering, where you can calculate metrics on your designs and decide which ones are good, which ones you'd like to test in the lab, and which ones you'd like to discard before you go any further. And so here, another thing I'd like to point out is that there's really kind of 2 paradigms for performing all of these steps. You have a more traditional paradigm of sort of physics-based design tools using things like Rosetta design. And then in the last few years, we've really been transitioning to more deep learning-based tools, which have actually really opened up the capabilities of protein design in general. And so today, I also want to point out that within these steps of protein design, when we think about moving towards DNA binder design, there's really several unique challenges in each of these 3 steps. In the case of backbone sampling, we need to be able to read out sequence of the DNA, not just through direct interactions with the bases, but also through shape readout, where every unique DNA sequence can have a sort of natural propensity for taking up slightly different shapes, like the width of the minor groove. And so we need to be able to create scaffolds that achieve this really precise shape complementarity to DNA, while at the same time precisely positioning the protein backbone in ways that when you perform sequence design, you can then directly read out the bases. And each base pair has a sort of unique molecular signature of hydrogen bonds, acceptors and donors and nonpolar interactions, which you can use to distinguish these bases. And here I'll point out that unlike in protein binder design, where you can often use hydrophobic interactions, we really need to rely a lot more to get direct readout through polar interactions. And that's quite difficult for the protein design tools that we've had. In addition, you have things like water-mediated interactions, which are very computationally expensive to model and are quite hard to do as well. So finally, in the in silico filtering step, we need to consider that DNA is a very flexible molecule. And so really any design model we're looking at is sort of one member of a conformational ensemble or spectrum of possible structures. And that can really change the way you think about how to do filtering, as well as the idea that you can have things like induced fit, where the protein binding itself can change the DNA shape. And so these are really what I see as the big problems in protein and DNA binder design. And so through my postdoc, we've been working on projects to start to address these things, and we've made quite a bit of progress. And I still think that many of these remain a challenge, and that's what I'll be continuing to work on in the next years. And so I want to talk today mostly about sort of a physics-based approach to DNA binder design and how we've slowly been transitioning to some of the more deep learning techniques. When I started my postdoc, really the deep learning methods that are becoming very powerful today weren't really mature. So there's sort of been this a journey of transitioning from these more traditional tools to deep learning tools as we've gone through this process. And we started off with this simple goal of designing small proteins, less than 65 amino acids, that would bind specific sequences of DNA, both as a model system for starting to develop these tools for protein nucleic acid interactions, and also because these small proteins can be useful for different things like designing new transcription factors. And so a lot of this was really inspired by work that had been done in protein binder design, where some of my colleagues, Longxin Cao and Brian Coventry, had published this really great paper where you could sample many different mini protein backbone scaffolds and look for those that have high degrees of shape complementarity to protein targets and extra interactions. And this had become a really powerful way of designing protein binding proteins. And so I thought we could just take this method and instead of having a protein target, have a DNA target. And so that's essentially what we tried out at first. But we started off with a structure of a DNA of a particular sequence. We can then go through a process where we create these comprehensive sets of disembodied amino acid side chains, what we call a rotamer interaction field or a RIF. And we target these to generate interactions with each of the 4 bases in our DNA target. We can then take a scaffold library and then dock these over this RIF in order to find docks that both contain some number of these disembodied side chains through positioning of the backbone coordinates, as well as having good shape complementarity. And what I mean is by not clashing with the phosphate backbone. From there, we have a dock, and we can perform our amino acid sequence design at that stage. And then once we have that, we can perform different filtering, basically calculating things like the number of hydrogen bonds across the protein-DNA interface and the quality of the scaffold, and how likely we think that particular amino acid sequence is going to fold into that actual shape. From there, we have a set of candidate designs, and then we go through a process of diversification, where you can either do this motif grafting process, where you take a motif from our design, like this helix interacting in the major groove, and then resample our backbones again over this in order to find new docks that can hold the same motif with these key interactions. Or we have this alternative method called inpainting, where we can essentially mask out different portions of the protein, like long loops, like shown in red here, and replace them with new protein variants that have different diversity. So these are just ways of generating more diversity. And so really, this is very similar to what was done with the protein binder design case. And I'll just say that for summarizing a few years of my postdoc, we went through many different iterations of this, and ultimately we're not able to generate any proteins that could bind specifically to a DNA sequence. And so really, kind of the innovation here was going through a process of trying to understand better how natural DNA binding proteins are actually working and trying to infer new design principles from that we could incorporate into our design pipeline. And so I'll just go through a few of these here. And so the way that we are trying to get sequence recognition was through a mode that's very dominant in nature, using a helix in a major groove that would interact with specific bases. And so what you see in nature is that it often uses many different orientations and positionings of these helices, each of which, with an amino acid sequence, can interact with a specific set of DNA bases. And so that's kind of one way that these proteins are able to achieve specificity. One of the things that we noticed about this is that the scaffold libraries that we had been using contained shapes that worked really well for protein binder cases, but were not really suitable for the case of DNA binding, because in many cases, we couldn't get many scaffolds that would bury a helix deep enough in the major groove without the rest of the scaffold clashing with the phosphate backbone or having opportunities for the right types of interactions with the phosphate backbone. And so ultimately, what we turned to here was to use scaffolds that look much more like natural DNA binding proteins, like these helix-turn-helix ones, which we ultimately created from AlphaFold 2 predictions of naturally mined helix-turn-helix proteins. And we just took the backbone shapes of those. And so here we're kind of taking a step back from a truly de novo approach to starting with these natural shapes, but still going through that process of de novo docking and sequence design. So that's really our first design principle, where we need to have proteins that position a recognition helix in such a way that it can have specific interactions with the DNA bases, while also having interactions that rigidly place that helix through interactions through the rest of the scaffold with the phosphate backbone. In particular, things like these backbone amine-mediated hydrogen bonds from ends of helices and loops to the phosphate oxygens. And from there, the next two design principles really came from looking at the specific types of interactions with the bases themselves. And so these are things that you often see in nature that we consider sort of privileged interactions. Things like these arginine G base bidentate hydrogen bonds, or similarly glutamine or asparagine to A base bidentates. And so these are things we see over and over again that can only occur between specific amino acid side chains and bases. And so we noticed that, and we also noticed that many of these side chains forming interactions with bases were also really pre-organized, and they were essentially backed up by other side chains in the protein, forming things like side chain hydrogen bonds and packing interactions that really fix them into place. And so one of the interesting things about that is our design tools, which are physics-based, designed with Rosetta, are really just trying to minimize the total energy of the system. And so they don't really consider things like interactions that would lead to specificity or things that would pre-organize these residues. And so we need to think about ways that we can incorporate in our design pipeline that would actually capture these types of interactions that would lead to specificity. So these are our next two design principles. First, we need to have things that would have these privileged interactions with DNA bases, as well as be able to pre-organize these side chains through side chain hydrogen bonds and packing interactions. And in all three of these cases, we can capture these through simple filters that we can just calculate on our design models, like the type of hydrogen bonds that are being made. And in the case of side chain pre-organization, we can use Rosetta's Rotamer-Boltzmann metric, which calculates the probability that a side chain would be in a particular rotameric state over all other possible rotamers for that particular side chain. But at the same time, we didn't necessarily want to just create filters that would select for the designs that were functional. But when we're performing sequence design on our docs, we wanted to make sure that every doc had its best chance of having these types of features that we were looking for. And so for that, we ultimately turned to revisiting the way that we were doing sequence design. And this is kind of the first step that we went from these more traditional physics-based design tools, where we're using Rosetta Fast Design for this sequence design method, and turn to a new network called Ligand MPNN, which is a message passaging neural network that's trained on natural protein DNA and protein small molecule complex structures in order to predict the most probable amino acid sequences given the backbone coordinates of the protein as well as the coordinates of all of the DNA atoms. And so this turns out to be a really powerful way of designing amino acid sequences in these protein nucleic acid complex structures. And another thing I'll mention here is that Ligand MPNN is much, much faster. That allows us to sample many more sequences in our design pipeline. So Rosetta Fast Design takes around 2 to 3 minutes per design trajectory, whereas Ligand MPNN can be completed and get you an entire amino acid sequence in around 1 to 2 seconds. So that was a big bonus as well. So here we did some benchmarking of this Ligand MPNN method versus Rosetta with the task of redesigning the sequence of native protein DNA complex structures. So with Rosetta, you get a sequence recovery in this case of around 35% of the native sequence. But with Ligand MPNN, we actually got a really big boost to about 51%, suggesting that it's starting to capture some of these more privileged interactions that you see in these native structures that are hard to get through just minimizing the energy with Rosetta. And Ligand MPNN is an extension of protein MPNN, which is a protein-only model that also does really well designing protein sequences in the absence of nucleic acid context. That got around 34% in these cases, which was interesting. And so now at this point, we can start to incorporate these new changes, this way that we're doing sequence design with ligand MPNN and these new filters that capture these different design principles. And it was at this stage that we started to get designs that were actually working in our method. And so this is still quite a hard problem. And so we ended up turning to high throughput screening to do this. So we essentially, for every DNA target, we would generate 5,000 to 10,000 designs computationally and use a yeast display assay to screen these. So we did this for 13 different DNA targets. All the proteins were synthesized in DNA and oligo pools ordered from Agilent. These could be transformed into yeast and then sorted for those that bound a biotinylated DNA when these proteins were displayed on the cell surface. Ultimately, from this process, we're able to identify 44 designs that bound to their intended DNA target, and these had various degrees of specificity. So here we're looking at a heat map of all of these 44 designs on the y-axis. On the x-axis, we have these 13 different DNA targets, and in red boxes indicates the intended target for each of these. And so what you can see is that some of these are quite non-specific, like DBP89, but some of these were really quite specific, like this DBP6, which really only had binding signal to its intended DNA target. So that was quite exciting for us to see. And from this, we were able to create a smaller set of around 5 proteins that were really quite perfectly orthogonal to each other, and each only bound to their intended DNA oligo sequence. When we looked at the affinities of these proteins, when we expressed them in E. coli and purified them, they tended to have binding affinities ranging from around 30 nanomolar to about 1 micromolar in KD. And here we're looking at the BLI traces, and we could see that in many of the most specific cases, we're kind of landing somewhere around 100 nanomolar. So that's quite a reasonable affinity that's comparable to many natural DNA binding proteins. And I think even more excitingly was the way that we could see how these proteins had specificities that really matched up really closely with their design models. And so here we're looking at 2 example designs. I was zooming in at the interface. You can see these nice hydrogen bond networks in our design models between the bases and the protein side chains. And then we performed this experiment, which is just a competition assay in our yeast display, where in addition to our biotinylated DNA target that gave us binding signal, we could add a competitor DNA of the same sequence and then scan through mutations of the competitor across all 4 bases at every position in order to find positions that broke competition. And so here showing up in red are positions that broke that competition, leading to higher binding signal. And I'm indicating with arrows the positions that were contacted with hydrogen bonds in our design model. What we can see is these positions that are most red tend to be around in the same positions that were contacted with hydrogen bonds in our designs. So that was really great to see. And you can also see how some of these more peripheral positions around those bases also had some degree of specificity, which is likely coming through some of the more indirect shape readout that we get and you often see in natural DNA binding proteins. And so this was sort of a more specific view of specificity in these designs, but we also did some looked at this in a bigger picture. So we performed some protein binding microarray experiments. And so here in these microarrays, you have DNA sequences that compose all possible 7-mer DNA sequences, and you can measure binding to each of the probes in the microarray, and from that calculate a value called an E-score, which tells you about the specificity of your protein to each of those 7-mer DNA sequences. And here we're plotting those E-score values for these 2 proteins for all 7 mers in gray, and the 7 mers that contain our designed target motif are shown in orange. And what we can see is that these tend to have much higher E-score values than the typical 7-mer sequence, really just showing that these do have a great deal of specificity to these specific sequences that they were designed for. And I thought that this was really quite cool because as compared to cases like protein binder design, where you're making binders to proteins with very different shapes that you can get specificity through shape complementarity. Here, it was kind of a newer case where we were able to design specificity for DNA, which has mostly the same shape with only subtle differences depending on the sequence. And most of the readout is through these direct hydrogen bond polar interactions with bases. And that's not something we've actually seen before in protein design. So that was quite a cool result. And so we were also able to solve a structure of one of these using crystallography. This is with the help of Barry Stoddard at Fred Hutch. And this was quite exciting to see that we had a crystal structure shown in blue overlaid with our design model in gray that actually matches quite closely between the design and the crystal. And you can see overall it's quite similar, but there are some differences, like in the way the site changes in the DNA shape, for example. And some of these differences suggest a lot of ways that we can continue to improve our design methods. Also, when we zoomed in at the interface, we could see a lot of these side chains that in our design model looked important for the binding are also showing up and doing very similar things, like the serine 39, as well as this arginine 38 residue that forms a bidentate hydrogen bond. You can see there's some changes in the rotamer, but overall it's in the same place and performing what looks like the same function. And you also see different things. So one thing I mentioned earlier is that water-mediated interactions play a big role in protein DNA. And even though our modeling steps not really able to capture these water-mediated interactions because they're very slow to calculate when we're going through many thousands of design trajectories at once, we do see that they show up in our designs. And so improving our methods for capturing water in our design pipeline is likely going to be an important way of improving our design methods overall. We also went through other ways of looking at the structural validation of these designs. So here we did a site saturation mutagenesis experiment to profile the footprint of the binder surface. And so here, every position of the protein was mutated to every possible amino acid sequence, and we could screen these all again with yeast display to calculate an SC50 value for every mutation. And here we can see residues at the surface of the protein tended to be much more amenable to mutation, but residues at the core or at the interface, as you would expect, are much less amenable to mutation. And especially residues like arginine 28 and arginine 29 really couldn't be mutated to much else, except for the case of R29, which had one mutation that was allowed. And that was interesting because although it still bound its DNA target, it completely broke specificity when we made that. So that residue was really important for the specificity of this particular design. And we were really interested in this design because when we had done these microarray experiments on this protein, we found that even though it had some degree of specificity to its target, we found that it had a really strong binding to an off-target sequence. And so that turned out when we did some Rosetta modeling, we found that this arginine 33 residue, which wasn't involved in the interface with the on-target motif, was actually forming this extra hydrogen bond interaction in the off-target case. And so when we made mutations to this residue, as well as a couple other positions that were interacting with the phosphate backbone, we actually found that we could dramatically improve the specificity from around 3 base pairs to around a 7 base pair stretch. And so what that means is that our design method is getting us really, really close to having an ideal sequence for a protein, but in some cases, we're just a few residues off. And incorporating things like specificity filtering against off-target sequences and negative design against off-target sequences could really help to improve these, because in our cases, we're really just designing for positive interactions with the DNA target and hoping that we get specificity through that. And so I think that's another area that we can really improve these design pipelines. And so at this point, we're starting to have the ability to design these small proteins that bind specific sequences, and we wanted to see if they could work in cells. And so we did a couple experiments to see if we could regulate transcription with these. And so the first one we did was to see if we could do this in mammalian cells with activation by fusing these small DNA binding domains with activation domains like VP64. One of the interesting things we found is that a direct fusion with VP64 was insufficient for achieving activation in these cases, and we needed to make an additional fusion with a dimerizing domain, this GCN4. So that brings multiple copies of these DNA binding domains together so you can get more affinity through an avidity effect. So ultimately with this, we were able to get some modest activation in cells with these proteins. And here in these different colors, we're just looking at different spacings between these two motifs that would each be occupied by half of the dimer. And some of those spaces worked better than others. And what I think was even more cool was what we did in E. coli, where we ultimately found that single domains similarly were unable to repress transcription in bacteria. What we ended up turning to is making structures that look very reminiscent to many bacterial regulators, where we use this software called Rosetta fold diffusion, which allows us to initialize these random backbone coordinates around two of these DNA binding domains arrayed around the DNA scaffold, and essentially denoise these coordinates into physically reasonable protein backbone structures that have these dimeric interactions, either in homodimeric arrangements that would have a palindromic target sequence or heterodimeric arrangements. And so ultimately, when we put these into cells, we could put the binding sites of these dimer structures in and around the -10 and -35 transcriptional elements in order to sterically block RNA polymerase, and we could achieve pretty good transcriptional repression with these systems. And one really interesting thing to me, at least, is that many of these, like these three here I'm showing here, all use the same DBP57 domain. They have the same specificity, but just by spatially controlling the positioning of these two domains, we could achieve orthogonal repression that would require different spacings of the DNA motif and orientations of that DNA motif. So I think that's a really interesting feature of design, that we can start to control these spatial positionings. And so with that, I just want to summarize this approach. We have this new method for sampling protein backbones and docks, designing their sequences, amino acid sequences, and then performing different filters using Rosetta physics-based modeling of the protein-DNA interface. And so this is quite exciting. For the first time, we have this ability to create new proteins that bind specific sequences of DNA. Many of these proteins are working quite well, some without any additional mutations, but in a few cases, we found that we could further improve these designs with small sets of mutations. And along the way, we learned these important design principles that helped us get this to work. And so all those things will help to inform our future efforts in this area. And so I think I may be running a little bit low on time here. And so I just want to briefly go through some conclusions and some of the current work that we're working on right now. And I may end up actually skipping a couple of slides here. But I just wanted to mention that we've been going through this process of transitioning from these sort of physics-based design methods, and you've already seen how we went from the Rosetta fast design method to ligand MPNN. But we've also been implementing a lot of new methods using deep learning to replace many of the other steps in these deep learning pipelines. And it's through this that in cases like protein binder design, people have in recent years been able to go from having to screen many thousands of design variants like we did in this case to only having to test a few dozen variants. So I'm really optimistic that by implementing these newer methods, we can actually substantially improve our success rates and make this a very accessible and easy to use method for designing new DNA binding proteins that would interact with specific sequences. And so to do this, we've been incorporating extensions of a Rosetta fold structure prediction network that predicts these protein DNA complex structures, and building on that, creating a new diffusion model that is essentially able to denoise these initialized backbone coordinates into reasonable DNA binding structures in the context of the protein nucleic acid docks. And so that, I think, will be a really exciting and general way of creating new protein scaffolds that can interact with DNA, both B-form DNA, as well as many other types of DNA or RNA structures that can be important to bind to. And so I think I'm just going to kind of skip the next couple of slides. These are just showing some of the complexes that you can predict with this Rosetta fold structure prediction network. And here we found that we were able to predict the specificity of many of our designed proteins. So this is not just working on native proteins, but also designs. And we also found that this structure prediction network could predict these dimer structures that we were able to create. So here in green is the design model, and blue is the Rosetta fold predicted model. So you can see it's really quite powerful at these cases. And so recently, we've been testing in the lab some designs with this entirely new deep learning-based pipeline where we start with this diffusion model, generate these new backbone structures, and design their sequences with MPNN. And then we've been able to confidently repredict these structures from sequence with Rosetta fold, and we can use that as a filter for our designs. And so I'm really excited to see how these proteins work in the lab. And I just wanted to end with this last slide, which is that we've really been focusing on just proteins that can bind specific sequences of DNA, but there's so much more that we can do in this area. we've started to design these sort of oligomeric dimer structures, but we can also start to think about making proteins that interact with DNA hairpin structures, proteins that start to manipulate DNA conformation with specific bends, and that can be useful for catalysis. You can think about getting cooperative effects where you have proteins binding to these distorted DNA structures in specific ways. And of course, we can start to go beyond just DNA and think about DNA RNA hybrids, RNA structures, and single-stranded DNA binding proteins. And so that's really kind of the world of possibilities that I'm hoping to open up through these new design capabilities. So with that, I'd just like to acknowledge everyone I've worked with, and many people have been contributing to this work I've talked about. Pictured here are some of the most important people, and also just want to thank my postdoc mentor, David Baker, as well as Frank DeMaio, who's given a lot of guidance in some of the new method developments that we've been doing. And so with that, I would love to take any questions.
Fantastic. Wow. That blew my mind. Oh, that's amazing. I want to ask Lynn if you have any question or comment to ask. While waiting, I mean, I'll start. So, oh, she's there.
No, just wow. That is phenomenal work, Cameron, and I'm certainly hoping that we can continue discussion offline.
Yes.
I can't imagine you'll have any trouble finding a job.
So do you have any comment or question to him, Lynn? Otherwise, I will start.
Yeah, I mean, these are really just, they're less questions and more thoughts of extending this to metallated base pairs and things like that, because you're well on your way. And also, I think I'm trying to wrap my mind around the idea of how this could be incorporated into astrobiology. Again, getting back to my closing comment that synthetic biology provides the art of the possible. You know, what were the initial DNA or even RNA protein interactions like? Because I think there's great interest in that. You know, were the nucleotides sort of, or the bases a structure? We find bases in meteorites even, but we don't find nucleotides. So, you know, it's a little bit of a chicken and egg, and I think that some of your technologies could certainly get at some of these questions. So again, fantastic, fascinating. I should leave it open to other people to ask questions.
Thank you.
Fantastic. Absolutely. So let me start with the one question, actually two questions. So I have the question entire time, but you answered some of my question by fusing your DNA binding domain with the ligand binding domain, right? Am I right?
Fusing with the dimerizing domain?
Right, the dimerizing domain.
Yes.
So if I remember correctly, a few years ago, Tanya Kotemi, I believe, also developed approach that de novo protein design for the transcription factor, basically. That's what I remember. But
Yes.
I try to understand the difference between your approach and her approach. Of course, from the same laboratory, you know, but she's a more, you know, old person, I guess. But one thing I want to, you know, ask is, you know, you, at the end of the day, ultimate goal, I guess you may have even everything kind of designed de novo, including ligand binding domain as well. In that way, you have the real transcription factor that is kind of bind to the DNA, but that also designed de novo. Also, the ligand binding domain also, you know, designed de novo. In that way, you have everything kind of artificial protein in some sense. So can you kind of tell us, teach us, especially me and other people, what the Tanya Kotemi, I think that science paper, I don't remember, but science paper 2021 or something like that, I don't remember exactly. What is difference between yours and hers? And then what we can do, kind of do what I just, you know, imagine.
Yeah, absolutely. Yeah, so I think what Tanya's lab did, which was Anam Glasgow's work, was a really nice way of designing protein ligand interfaces where they were able to design sort of chemically inducible dimers for a particular ligand, which I believe was farnesyl pyrophosphate, I think.
Right.
and which is an important molecule in metabolic engineering things. And what they were able to do is to get transcriptional regulation by having each half of the dimer fused in a flexible way to a DNA binding domain. And so that can get you sort of this dose dependent titratable transcriptional regulation. What's different in my case is that, well, for one, we haven't really got to the part where we can have small molecule binding, but in many transcriptional regulators in nature, you have this much more switch-like function where you coming from the allostery, which I think you can't really do with the chemically induced dimer system, where you're going to have a more like even titration. And so what I hope to be able to do eventually is to create things that really look a lot like natural allosteric transcription factors, where you can be bound to DNA and then have small molecules binding, and then having really subtle changes that lead to switch-like changes in transcriptional regulation. So I guess in a sense, it's kind of a subtle difference, but I think a very important one for a lot of engineering, a lot of biological properties and things like gene circuits.
I see. So did they also make the DNA binding domain de novo, or they only focus on the ligand binding domain only?
In this case, it was the ligand binding domain only. Yes, and
So in that case, I mean, your work combined with her work, now potentially we could make the real everything just de novo, including DNA binding and ligand binding together.
Yeah, absolutely. Yeah, I think eventually combining these things will be the way to go.
Okay, so that's what I kind of envision. So somebody wanted, and I want to see somebody just do it. I mean, probably that's you, I guess, in the future. My second question is, this is amazing. We now have the de novo design protein bind to one of the most complex, molecule, macromolecule. But I want to go the completely opposite direction. So Lynn also, indicate earlier, how much nature nature do a lot of great thing, evolution doing a lot of great thing, including binding to the one of the smallest element, that's a metal ion.
Mm-hmm.
So have anyone kind of done making de novo protein that binds specifically metal ion, for example, differentiate between copper ion versus ion, the whatever other metal ion, or there are element that's one of my interests, in the future.
Yeah, absolutely. So this is a very active area of research among many of my colleagues. And so there was a paper recently where there were some metal ion binding proteins using that type of diffusion method that I mentioned. That was really exciting. I'm not totally sure how much specificity they have in that for specific ions. I think there may be some degree of specificity, but there's a lot of work. Many of my coworkers are working on this exact problem right now.
In your laboratory?
In the David Baker's lab, yes.
Okay, great, wonderful. That is one thing I want to see, because I wanted to develop something for the rare earth element especially. But I know, the natural protein already exists for the rare earth metal, but the specificity is not there yet, because that's a tiny metal ion. How could you differentiate between the very tiny thing? That's kind of something I want to see in the future. Wonderful.
Yeah, if I could add,
sure, please.
If I could add a comment, for the biomining, I did have a PhD student doing a little bit of that work with copper, and not with the same sort of sophisticated modeling that Cameron's using, but in the end, the naturally evolved motifs were actually the most effective. And maybe that's sometimes nature wins out. Nature's had over 4 billion years head start on us.
Yes.
And sometimes we can do better, but
yeah,
you're certainly right. The rare earths become incredibly attractive and interesting, and needs to be done.
Yeah, so I'm not expert in the that type of de novo protein design, so I also rely on sort of directed evolution or some subtle change using the already existing protein out there, and that's what I want to do future. Or Cameron may help me. Once he set up his laboratory collaboration. Fantastic. Any other thing? Let me check whether, okay, I didn't see any question except the question that is asked, but Lynn already answered that question online. So let us close because we now see 11:04 my time. So thank you all for joining and staying today. We'll meet again February 4th, already February, next week. The same time, the same Zoom link. We'll have Professor Chris Rao at UIUC and Dr. Christian Meyer at University of Colorado Boulder. As usual, the follow informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks. I'll stop recording. Just give me 1 second.